Yeah, so I'd like to introduce you to Jason, who will talk about efficiently detecting interaction from high-dimensional observations of pairwise perturbations. So Jason is an incoming fellow at the University of Manchester, starting in October 2024. So maybe, are you taking students? Yes. Anyone looking for a PhD is your man. And he's also a research unit lead and staff research scientist at Valence Lab. Previously, he was a postdoc with Joshua Bianchio at University of Montreal. And before joining Mila, he was a PhD student at the University of British Columbia with Kevin Nathan Brown. He works on developing techniques for causal representation learning and causal inference. And a special thank you to Jason for filling up that spot last minute. But he's a great presenter, so I'm sure he has everything ready.
Great. Thank you so much for having me. Yeah, so this is all pretty last minute. So if you spot some typos on the slides, tell me and I'll be nice and embarrassed. Okay, so my talk today is going to be maybe a little different from what we've seen so far because it's thinking more about the observations than what the perturbations are themselves. So I know mobile's primarily about thinking about molecules. And here we're thinking more about well, how do we measure the effects of molecules? And sort of what is specifically in these high dimensional observations. And the main motivation for this is we can collect, these unstructured measurements of cells very cheaply, right? And we can collect these measurements tend to be really information rich, and they're relatively easy to incorporate into these highly automated pipelines. So I've spent the last year at Recursion, or at Valence, which is connected to Recursion, where they have these sort of amazing robotic labs for imaging cells at scale. And so that you can really collect a lot of data. But the data you get out is this sort of unstructured image data, which kind of presents challenges, and that's what we're going to be talking about today. First, kind of to motivate what I mean by information rich. I find this kind of fascinating that if you choose the right embedding of these measurements, you can get sort of surprising amounts of information about the underlying biology. So in particular, if you take kind of cosine similarities between embeddings with a really large model, and you look at the cosine similarities between sort of genes on the same pathway, you'll find that they tend to be highly cosine similar. And this is maybe obvious in retrospect, but I found this surprising when I first saw it. The idea is if you have sort of two genes that are coding for proteins that form a complex, I knock either one of them out, I've disabled that complex. And so I'm going to see similar morphological features in sort of image space. And so maybe it makes sense in retrospect that they should be highly cosine similar. But I still find it surprising that we're sort of reasoning about things that we can't see visually, in the pixels in the image, but we just sort of can reason about this from the sort of morphological changes to the cell. Okay. But all of this is operating at the level of single perturbations. So cosine similarities will tell us kind of the effect of individual molecules or gene knockouts on the cell. But if we want to sort of start to think about combination therapies or extract any kind of causal understanding of what's going on within a cell, we need to find a way of getting past sort of single perturbations. And this is a problem because we can scale to single perturbations, but scaling to even pairwise perturbations is really, really hard, right? The combinatorics are against us. If you wanted to knock out all pairs of genes on the human genome, that would be on the order of 200 million experiments, and that's to say nothing about kind of libraries of molecules where you might want to do pairwise molecules, and that's just way bigger than any experimental lab can do. And so this becomes an active learning problem. Essentially, you want to choose pairs of perturbations that allow us to find kind of interesting drug combinations or interesting pairs of perturbations. But it's kind of an odd one because we're getting measurements in image space rather than measurements in the sort of traditional low dimensional space, right? And so kind of the more fundamental question in some sense is if our experimental outcome is an image, how do we even define what it means to be interesting, right? What are we searching for if our image outcome is an image? The basic mantra that we've taken in this work is that we should experiment where outcomes involve unpredictable interactions. We're going to spend most of the talk defining what we mean by that. And then we should just generalize to the rest, right? So machine learning tends to be really good at generalization. Let's kind of generalize where we can and experiment where things are unpredictable. And what I'm going to argue is that the surprising implication of this approach is that this can tend to lead us to experiment on things that are actually biologically interesting as well, right? And that's kind of the part that is more surprising, right? Okay. So how do we reason about, or how do we think about this problem? Well, the way I tend to approach this problem, coming from a causal inference background or causal representation learning background, is we think that the cells that we're imaging have some latent state. We're not going to specify what that is. We're just going to say there exists some latent state that describes the cell, which we use Z to denote. And then the images we observe are sort of pixels in an image that are sort of some rendering of that latent state, right? So we'll use the function G to describe the process of imaging the cell into pixels. And we can think of this as like a high-dimensional kind of readout of the underlying cell state. And then we're going to do perturbations on these cells, right? So we're going to perturb them by knocking genes out or applying different molecules, and that's going to push around this latent state by deactivating pathways or interacting with molecules and so forth. Okay. And the main assumption we're going to make is that the pixel level information is independent of these perturbations if we were to know this latent state, right? So kind of the latent state is capturing all the changes. We're not doing anything. We're not affecting pixels directly. Okay. And then the question we're trying to ask is sort of the most basic question you might want to ask is do two perturbations have independent effects on the cell? Okay. Now this is a question that kind of is you asked frequently. So in drug discovery, you'll ask things like, do you have synergistic or antagonistic relationships between molecules? And usually this is measured using something called a Bliss score, which I'll explain in a minute, which talks about sort of independence in terms of cell death. In biology, they do similar things where you're interested in whether pairwise knockouts of genes lead to so-called synthetic lethality, which is where cells die at a rate that is unexpected compared to what you would have expected from single perturbations on their own, right? So if I knock out gene 1, maybe the cell's fine. Knock out gene 2, the cell's fine. If I knock out both, the cell dies. Could be an example of synthetic lethality. And they measure this using independence tests on cell viability, right? Now, at its core, these two things are basically testing the same thing. Maybe it says something about people working on drug discovery and biology that one talks about cell death and the other one talks about cell viability. Optimist, pessimist. But really, it's the same independence test, right? You're measuring, if you're measuring the fraction of cells that survived, you're measuring this kind of classic, something you would have seen in first year statistics, which is, can I move my mouse? Never mind. Which is just a product of two Bernoulli probabilities. If the two things are independent, you've got just a product of probabilities of Bernoulli random variables. And if not, so you're checking whether the fraction of cells that died in the double perturbation is equal to the product of the fraction of cells that survived in perturbation 1 and the fraction of cells that survived in perturbation 2. On the other hand, if you had chosen to write down the number of cells that died instead of the number of cells that survived, you're checking an or relationship because you could be either killed by perturbation 1 or killed by perturbation 2. And in which case, you're comparing in the double knockout perturbation, you measure some fraction of cells that died, and you're comparing that with the sum of the fraction of cells that died in perturbation 1, fraction of cells that died in perturbation 2, minus the products of probabilities to get rid of the interaction term. Okay, so this is very simple first year statistics stuff, but I guess the thing that's sort of interesting to think about here is when we're in control of this measurement process. So in this case, the state of the cells are the same. The things that are sitting on a petri dish are the same, and what we've chosen to write down changes the test we run, right? If we've chosen to write down the fraction of cells that survive, we run one test. If we've chosen to write down fraction of cells that die, we run a different test. And the challenge here for us is that now we're going to work with pixel space, right? So we're not in control of that measurement process anymore. Okay, so we're just measuring pixels in an image. And sort of, can we still answer the same kinds of questions? Okay. The main idea that I'm going to show is that we can still answer these kinds of independence tests by looking at how changes in conditional densities are affected by perturbations, right? So why does this work? Well, I said before we had this latent distribution. So we have these sort of, we're talking about cell state in terms of this some distribution over latent variables. And if you remember the change of variable formula, that's good. If you don't, just as a reminder, we can express, we said that the pixels that we saw are some function g that is a transformation of this latent state. And the change of variable formula tells us that we can express this. We can express this conditional, this density over x as the density over the latents times this absolute value of the determinant of the Jacobian, which is a mouthful, but fortunately, we're going to get to kind of cancel that term out, so you can largely ignore it for our purposes. And so we can kind of talk about what we're going to be trying to do is talk about the changes in conditional density in terms of this changes in latent state rather than in terms of pixel space. Thank you. That's great. I can now point to things. Okay. So for simplicity, let's assume that each perturbation only affects a single latent variable. It's going to make it easier to explain. And furthermore, that this latent variable Zi is a leaf node, right? So if we were sort of describing the causal relationships between these nodes, we're going to assume that we're working with just one of the leaves. And then we can sort of write out this latent distribution as just a product over the distribution over any given latent variable time given its parents. And when we do a perturbation, we only affect this leaf node's distribution, right? Because we've assumed that Zi is a leaf node, and we're perturbing only, and kind of by the causal factorization, we're only perturbing one of these terms in this joint distribution, right? And so so if I look at the density ratio of the perturbed cells to the control cells, it's going to have a nice simple form under these assumptions, right? So I can just compare the perturbed to the control from what we sort of saw on the previous slide with the change of variable formula. We have this is just literally writing out the change of variable formula, and all of this, all of this stuff on the right here is going to cancel, right? Because that's all the same. And so all you're left with, the sort of change in distribution that you're inducing with a perturbation, is just a function of the sort of variable that you've perturbed, the latent variable that you've perturbed, and nothing else under these assumptions. Why is this useful? Well, it lets us test for independence of effects by testing whether the product of these density ratios equals the density ratio of the double perturbation. So sort of exactly analogous to what we had seen with the sort of Bernoullis, we can now do this in terms of density ratios. And so we can see this over here. So on the previous slide, we just argued that this density ratio for leaf nodes is going to be equal to that. Similarly, this density ratio for another leaf node would be equal to that. And if these two things are independent, then you're just going to have the same thing on the right-hand side, right? And so this is sort of like, if you have independence, you have this nice relationship that is testable. Okay. Okay. So that gives us kind of a signal for when two perturbations are independent. We now want to turn this into something analogous to a test statistic. Because this is just something we can evaluate on an individual image, but it's not going to tell us what's happening at a distributional level. So we want to turn this into something like a test statistic. And full disclosure, we're sort of still actively working on this, and there are a number of design choices that you could make here. A simple thing you can do is just taking the expectation with respect to the control distribution. So this is just evaluate this expression on samples from the control distribution. and then if you just sort of write out that, so then, and we do this in log space rather than the sort of original density ratio space, then what you essentially have is testing this relationship of KL divergences. Okay, so just kind of plugging in this expectation over density things, doing it in log space, you're essentially testing this relationship between KL divergences. And so this then becomes a test for independence, which we can then use downstream. How am I doing on time? All right, good. Okay, so that's one notion of independence. I'm going to tell you about a second notion of like independence, which I will be the first to disclose is a much weirder one to think about, but it has a nice interpretation in terms of composing embeddings, and so it's worth looking at. So rather than testing independent events, we can test disjoint events. These are sort of two different notions of non-dependence. By testing whether the density composes additively, okay? I'm not going to go through the details, but this is very analogous to what we saw on the previous slide. But whereas in the previous slide, we were talking about whether log densities composed additively, here now we're just talking about whether densities themselves compose additively. So sort of if you have this relationship between densities, then we can show that any embedding that you have of these cell images will compose additively in a way that you would like. So sort of in a way kind of analogous to kind of word vector addition that we've all seen in machine learning context, right? So if you've ever seen the sort of man is to woman, king is to queen kind of relationships that come up in word vectors, you can do the same thing sort of under this distributional assumption. You'll get the same thing with embedding. So the difference between the control embedding and perturbation of gene 1 and the control embedding and the perturbation of gene I and gene J, summing these up will give you the double knockout, right? This is a strange distributional assumption, and you know we still don't feel like we understand it well enough. But this fact that you can do this for a lot of pairs of perturbations shows up a lot, right? And there's a growing set of empirical evidence that we're able to compose embeddings of this form for a lot of perturbations, not all perturbations. Clearly, you know, interesting interactions are the things where this fails is where things are interesting. But there's a sort of surprising empirical observation that this shows up a lot, and this gives sufficient conditions for this to happen, right? We can test whether this holds explicitly. Yeah. Oh, sorry. Okay. So this relationship that I've said holds for all embedding functions H, right? So it's not sort of particular to some particular choice of embedding function. This will just hold generally. But that includes sort of trivial cases like an embedding function just sets everything to 0. If you want to instead turn this into a test for whether this distributional assumption holds, you want to search over all possible embedding functions and try and find the kind of the worst case one and see whether it still holds there. And that ends up being equivalent to what's called a maximum mean discrepancy test, which is just a test for independence of test for test whether two distributions are the same. And it's essentially these two distributions. That's equivalent to testing these things. Okay, so we can show this as well. Okay. So sorry, that was a lot of math, but we're now going to switch gears into results. So first, sort of just simple synthetic examples for whether these kinds of tests actually show what we would expect when we know the data generating process, and kind of indeed they do. The KL divergence thing is more sensitive than we would like, less sensitive than we would like. We're not seeing all the dependence showing up as strongly as we would like, and so that's why we're sort of actively working on different methods. Whereas the MMD thing is very clearly kind of detecting dependence where we expect dependence, but we're actively working on improving these methods. We then went into this on real data. We've done this for all pairs of 50 gene knockouts, as well as all pairs of 50 compounds. So you're sort of looking at compound combinations and then also gene compound pairs. We're gonna look at genes in these slides, but we have results on the compounds and the gene compounds as well. And so it's this on the right here is MMD. So that's the thing I showed previously. And actually, interestingly, you get sort of a pretty similar pattern of interactions if you just look at the final hidden layer of a classifier. And there are reasons for that, but I won't go into that. Okay, so I'll show you sort of that on its own isn't that meaningful. It's just like a pairwise plot. But what's interesting is to then go and look at sort of what are the highly interacting pairs. How do they? Can we say anything about what seems to be interacting strongly? And this is a plot which I'll be the first to admit is not the most interpretable plot to read, but it's the thing that kind of got me interested in understanding what was going on under the hood when we first made this plot. So let me try to talk you through it. On the x-axis, we have the cosine similarity thing that we saw on the first couple of slides, right? So this is sort of how recursion finds interactions in the sense that we talked about before. If things are highly cosine similar, so like points out in this region, we'll say they're likely interacting because they have similar morphological effects, right? So they're probably on the same pathway. So that's the sort of cosine similarity metric. And then on the y-axis is this difference in embeddings, right? So I said things are going to compose when they're independent, and when they're not going to compose, right? And so anything on the x-axis is kind of cheaper to collect because it's just single perturbations. So you can compute these things from single perturbations. Whereas on the y-axis these are double perturbations, so they're much more expensive. It's going to motivate the last part of the talk, but it's showing up. Different things are showing up under these metrics, right? So these are sort of outliers in terms of single perturbations. These are things we would have called interactions under the original test. These are outliers in terms of the norm difference of the embeddings. And so these are things we're going to call interactions under these new metrics. And then this is stuff that would show up in both metrics, right? So now evaluation is hard, but what's interesting is the things that show up as highly interacting are things like pairs of genes on the apoptosis pathway, on the proteasome, whereas things that are showing up on over here are things are like on the autophagy pathway. It's at least kind of consistent with what we know about biology that you should expect more redundancy on the apoptosis and proteasome pathways. These are things where you don't want kind of some malfunctioning protein to cause the cell to kill itself, which is the apoptosis pathway. So it's like these are inhibitors, and so and so if you're looking for kind of synthetic lethality kinds of relationships, finding sort of things. Finding these pairs of perturbations being strongly correlated with the apoptosis pathway makes sense. That said, I mean, evaluation of these things is extremely hard because we're going to detect interactions that go beyond what is known in biology because we're measuring interactions in kind of changes in morphology space, which is more general than just cell death, right? So you can have sort of changes in morphology that don't lead to cell death, and so many of the relationships we find will not be already known in biology. Cool. Okay, so this is all fun, but pairwise perturbations, as I said at the beginning, don't scale, right? And so it's in some sense academic because we can do this for pairs of 50 perturbations. How do we do this for the whole genome and the like? So this takes us back into the world of active learning. And I'll note that sort of like active learning approaches are often derived from kind of an information theory perspective in terms of finding examples that maximize information gain under some notion of information gain, right? This perspective is really challenging to apply in these settings because when your experimental outcomes are images, kind of every observation is information rich. And so you're kind of comparing high entropy things to high entropy things. Good estimates of entropy are hard in themselves, and so like reliable comparisons between these high entropy things are very difficult. But we've just derived a bunch of test statistics that kind of tell us where things are going to interact, right? So we've done all this work to derive these test statistics that kind of give us an indication of whether two things are interacting. And maybe to kind of drive the point home, you know, if you think about just like looking at images like this, it's not at all obvious whether two perturbations have interacted or not. The cell dies maybe, but in general, you know, like I've seen one perturbation, it changes the morphology slightly. I see another perturbation, it changes the morphology slightly. Have these two things interacted? I don't know. I'm just seeing a third, you know, I'm seeing a double knockout which has a small change in morphology. So even defining what it means to be an interaction is not obvious, but we've just done a whole bunch of work to define that, right? So, and as a result, we've got like a set of test statistics that in some sense define a reward that we can use, that we can aim to maximize. We want to find places where things interact. And these will be the things that are unpredictable according to our test statistic, okay? And so what this does is it lets us reduce the problem of like active learning from images, which is a really hard thing to think about, to a problem of active matrix completion, where we're sort of sequentially selecting experiments to run in this sort of pairwise matrix, right? And we've got a score for every pair of experiments. And that's a much better, well-studied problem. Okay, so the approach then is we're going to learn a reward model that's going to predict this test statistic for every perturbation, every pair of perturbations, right? And so in this context, this is literally just matrix completion, right? So kind of inductively, let's assume we've selected some set of pairs of perturbations. Obviously, this works with when we haven't done anything, but it's easier to think about once we've got some of this empty matrix filled in. And we can do matrix completion to kind of predict the rest, right? And then at every round, we're going to select the index that minimizes instantaneous regret per bit of information gain. That's a mouthful, but essentially this is a way of trading off exploration and exploitation. Okay, so we have our reward model that has some posterior over how likely we are to see an interaction. In particular, it's like a posterior over the test statistic for all pairs of perturbations, and you could sort of query it at every point. You know, some points will sort of be pretty certain that there's going to be an interaction. Some points will be very uncertain. And then what we want is points kind of like this, which are points where maybe we're going to see a strong interaction, maybe not. We're very uncertain. Querying that point will give us a large amount of information gain. And if it happens to be a good point, we'll select that point. Okay, so, you know, that's and using this approach, we can very efficiently find interactions under our metric, right? So, so this is this on the left here. We're looking at the percentage of the highest interacting pairs that we found over rounds of experimentation, right? So we sort of like take the top 5 percentile of test statistics because we know all the test statistics in this experiment, and we see how quickly we find all of those in the top 5 percentile. And this information-directed search approach that we looked at on the previous slide tends to find these things very, very fast, faster than things like Thompson sampling and UCB, and certainly a lot faster than random search. In terms of known interactions, so we can also evaluate with respect to like how many of these interactions are actually known biological interactions. The differences are smaller, and this is kind of in part due to what I was saying before, is many of the interactions we're going to find are either false positives, like it's totally possible that we'll have false positives because there are some distributional assumptions that we've used. If those assumptions are wrong, those are going to lead to false positives. But alternatively, they're just novel biology that hasn't been discovered before, right? And so we really have to go and do some orthogonal assays to tell the difference between like this big difference versus this much smaller, more modest difference. Here's a fun animation of what the search procedure looks like. So this is sort of over experimentation. Random search, you can't see this that nicely, but random search is just obviously going to sort of randomly explore, whereas information-directed search is going to kind of like pick out these highly interactive genes and do more experimentation on those rather than on than doing it randomly. And so that's what explains the big gap between these two things. Okay, that's all I have time for. I just want to say thank you to to all the various collaborators on this. Most of the theory around detecting interaction has been driven by David, who's in the audience here. The active learning parts was driven by Moksh, who's also in the audience somewhere, and Anikit. Sean and Ali played a big role in trying to stare at these results and trying to understand the biological significance of them. And then Burton has been with me on this project from the start and helped set up the experiments, as well as been providing support throughout. And then finally, sorry, just some concluding thoughts. I think what's nice about this project is it kind of marries ideas from causal inference and causal representation learning, which allows us to kind of reason about where we can generalize and where we don't expect to generalize, and then design our experiments to go where we don't expect to generalize. I think it's interesting to think about how this generalizes to higher order interactions, so things like molecular fragments. We can probably think about in the same framework. And then as a sort of aside, this is more to my own community, the causal representation learning community. I think this work kind of presents a different perspective on causal representation learning. Much of the work in that community, including some of the work that I've done, focuses on explicit disentanglement. And that's a very challenging thing to do in practice, because you can't validate whether you've successfully disentangled something, right? So we see methods that seem to work nicely on synthetic problems, but then when you try and apply them in these complex high-dimensional settings, they can be very challenging to work with, because you get an estimate, but you're not clear if it's right, and it's very difficult to test that. I think what's nice about the testing perspective, so we're rather like kind of falsifying distributional assumptions rather than trying to disentangle, is it lets us, if we make incorrect distributional assumptions, it's just gonna lead us to false positives. But this is something we can then go and test with orthogonal assays. And it also just makes it far easier. We can actually validate our machine learning methods with actual explicit validation sets. This is very important when you're trying to fit these high-dimensional models, and it can be very difficult to do in standard disentanglement approaches. And so, yeah, so the thing, kind of this research program that I'm excited about is kind of thinking about like how observable quantities that we can estimate, like these conditional densities, can be validated and allow us to kind of extract the causal relationships that underlie these observations, these high-dimensional observations. Yep, that's it. Thank you.
All right, thanks a lot for the very interesting talk. Is there any question? Please come to the microphone.
Thank you for. Hello. Thank you for your talk. Could you go back to one of the first slides when you introduced the diagram, the DAG structure?
Yeah.
Yeah. Here. So, trying to understand why the arrows are not the other way around, at least between X and Z.
Between X and Z.
Yeah, because I mean, so there's two things. First, like Z is a latent state.
Yeah.
This is an observable.
Let me try and see whether. So we're thinking about the direction of information flow, the kind of causal direction of information flow. So the cell lives on a plate, and then we are imaging it. Like there is sort of light that gets, this function G is kind of measuring the process of like imaging that onto pixels, and so that's why the causal direction is in that direction.
So that makes sense. But okay, what I'm trying to understand. So you're assuming that the latent state partitions into a set of Z variables.
Yeah.
Like I'm wondering how reasonable it is to make this assumption in practice. We have a cell that, for example, or that kind of has a state where all these things are interacting with each other. Like I'm wondering if the partitioning assumption makes sense.
Good. Okay. So this is a great question. It's extremely subtle to think about these things. The way I like to think about latent variables in this context is as kind of morphological features of the cell, right? Things that you could point to and say, ah, that cell is dying. So it's it's clearly a dying cell. So we have a sort of latent variable that describes that state of the cell. That cell is experiencing autophagy. There's some morphological features associated with it. These things may be interacting, and these are kind of the output of pathways that exist underneath it, which we can't directly observe. Much of the theory in causal representation learning depends on an injective relationship between latent variables and observations. And so, if you could in principle say, well, the latents are the genes, or the latents are the proteins in the cell, but that then you clearly don't have an injective relationship. These we can't with a microscope see what proteins are active in the cell. We can only see their sort of morphological effects. But this is extremely subtle, and we could have a long conversation about it. So maybe.
One more question. In the later slides, you had an H function that you were optimizing over.
Yep.
You had a supremum over it.
Yep. I know this. Okay. So that's.
I was wondering what was the family of functions H that you were sort of optimizing over
Yeah.
in practice? Is it like linear functions?
So it would be kernel. It's a kernel method. So it's so you're looking at like RBF kernel, space of functions that are defined by RBF kernel, Matern kernel, or the like. So it's the reproducing Hilbert space.
So you're optimizing the kernel in that case.
Yeah. So it's just a test, but you're basically looking for a test function. You're looking for worst case test.
Does this guarantee that it's actually sufficient to have if you have all the expectations in that?
So
then it applies for the density function.
Yeah. So this will work if you've got if it's kind of a rich enough family of functions, basically. So you need a kernel that is recursive enough. Yeah. So yes, and if you're prepared to assume that.
Thank you. That's it.
No worries.
Hi, nice talk. I was wondering if your computational framework was able to differentiate between two types of genetic interactions. There are like positive genetic interactions, which are between synergistic genes, and there is the other type, which is between antagonistic genes. So I wonder.
Yeah, that's a great question. So sort of can we? I'll just repeat the question because Mike's a bit soft. But can we tell the difference between a synergistic relationship and an antagonistic relationship, or the like? The short answer is I don't know how. The long answer is I think that it should be possible by combining what we observe from a single perturbation, these sort of cosine similarities, and these scores. So kind of combining these different scores. So single perturbations will say, if I've got sort of like two single perturbations that are very highly cosine similar, those are having like similar direction effect. But then if they're also having a very surprising interaction, that would then suggest that there's like an antagonistic relationship, because it's like you would expect these things to compose, but in fact, it's sort of they don't. That kind of thing seems plausible, but we haven't worked it out yet.
Another question. So.
Yep.
You touched upon redundancies, functional redundancy. It's very well known that duplicate genes, there are lots of them. They are enriched with synthetic lethality. So I wonder how much proportion of.
Could you speak up a little bit? Sorry, I'm just.
How much proportion of synthetic lethal interactions that you found are between duplicate genes, like redundant genes?
That's a good question that I'm not sure about. So we didn't originally, when we chose the 50 genes that we knocked out, we chose them before we decided to look at synthetic lethality as being something of interest. And so there are a couple of synthetic lethal pairs in that set, and we do find that they tend to have large scores under these metrics, but we don't have like a set of genes that we know, like a large set of genes that we know are all synthetic lethal for the pairs. And so it's sort of hard to answer that with any kind of confidence.
I think great talk, especially the push for more testable implication of possible representation. I think it's a great direction. If you go to the slide with the active learning benchmark.
Yes.
This one. For example, the random sampling, for example, like yes. What kind of test function was that? Was that real? Well, it's by.
Sorry, this one?
Yeah.
Yeah.
So what's on the left? What kind of test function is that?
Over there. When you say what kind? So this one was using the final hidden layers of the embedding function. So this is testing the non-interaction, so the disjoint setting. We're busy actually reproducing these figures for the MMD test and for the KL, but we haven't done those yet.
And on the right here, the confidence is being higher because anyway.
Yeah, that's a good question. So like there's a sort of big overlap in I think so the right-hand side is this percentage of known interactions. Like I want to report it because I want to be upfront about it, but it's subtle because of this problem of like evaluation. Like really the thing to do is we see some interaction. We should then go and say do some orthogonal assays to see whether these interactions are real or not. Kind of evaluating us with respect to known interactions, you would hope we find known interactions, like that you would expect, but like we will find many other interactions which we say are interactions, and even if they are real interactions in a morphological space, they may well not be known in biology. And so yeah, I would expect like false positives are going to make us look bad on that metric, and sorry, false negatives are going to make us bad. So they're like they're real relationships, but they're just not known to biology. Will look bad on that metric, and so it's hard to tell kind of how much of these are false positives versus just bad metrics.
Just looking at it, what color is UCB? Is it green? Is that worse than random?
No. So random was the that is weird. Sorry, that UCB was definitely better than random. So that seems, I think this might just be rendering weirdly. Oh no. Okay, no, that is very strange. I was having a look at it. Yeah, I would expect UCB to be better. Cool, thank you.
Do you have a question? Do you have a question or no? Okay, there's no more space. Okay, thanks a lot for the talk. If I may also ask a question. So you show that you can discover like when there's an interaction between the genes by looking at whether the sum of the vectors correspond like to the actual effect. But I'm wondering, like, especially when you consider that in many cases these interactions are like, okay, well, the cell resisted to one intervention, but when adding a second intervention, then like the cell died, for example. Like, do you think that the effect that you would observe on all single genes would be visible enough from like the batch effect, for example? Like, I think like if you were to test to take this the same cell and like clone the clone the universe and reproduce the same experiment,
yep,
to make a lot of sense. But considering that there's so many noise in the experimental setting, are you actually like, how confident are you that you can really take these vectors and sum them up or subtract them to retrieve interactions?
Yeah, that's a great question. So, I mean, I think to deal with batch effects, we end up having to do a lot of averaging, and that's true of both the cosine similarity relationships and the things. So, but if you look at, I don't know, this plot isn't great, but if you just stare at this plot, you can see that we get sort of close to zero predictions for a lot of things. For a significant part of this matrix ends up being close to zero. To do that, you have to do averaging over a number of different guides and a number of different replicates in order to sort of average over many of the batch effects, and then you start to get more reliable signal. But even so, like, you can certainly have the case where a perturbation that has a very minimal effect on the cell could then end up having sort of point in random directions, and then will not be a reliable signal. I think the right way to deal with that is just having a good kind of pre-processing step that is checking whether you're getting reliable perturbation consistency, and then only make these conclusions on things that are sort of reliably perturbing things.